In this video, we're gonna talk about renal anatomy. Each kidney has an outer cortex or the renal cortex, which projects toward the medulla, and this part of the cortex is called the renal columns or the columns of Berdini. And these are the renal medulla. Renal medulla looks like a pyramid, so these are also called the renal pyramids. Each renal pyramid has a base and a renal papilla. At the tip of the renal papilla, there are small openings. These are called the foramina papillaria. And this perforated area is also known as the lamina cribrosa or the cribriform plate. And these striated structures are called these are the medullary there are the striated structures actually consist of tubules that drain the urine separated from blood in the renal cortex and transfer toward the medulla. These are the stria medullaris. Stria medullaris. In the kidney, this the renal pyramus and surrounding renal cortex together make up a renal lobe. And this foramina papillaria actually are the openings of the collecting ducts. This is a papillary collecting duct. Through the collecting ducts, urine empty into this minor calyx. Minor calyx caps the renal papilla. This minor calyx is a minor calyx, which unite and form the major calyx. Major calyxes unite and form the renal pelvis. Renal pelvis, as it goes down, becomes narrowed and then continues at ureter. This is the ureter. Now, there is another structure, it's called renal sinus. We should not confuse renal sinus with the renal pelvis. Renal sinus is the part of the kidney which holds the renal pelvis, renal vessels, and the fat tissue in it. Now, if we take this out, the space that houses these structures is the renal sinus. Here is the renal hilum, through which the ureter and the renal vein, leave the kidney, but the arteries, renal artery, and the nerves enter the kidney. In the renal hilum, from anterior to posterior, we have the following structures. From anterior to the posterior, we have the renal vein, then two branches of the renal artery, then the ureter, then the third branches of the renal artery. Now, this is the renal artery. Renal artery branches into segmental artery. Here is the segmental artery. Segmental artery divides into lobar artery. Then lobar artery continues as the interlobar artery. This is the interlobar artery. Interlobar artery becomes the arcuate artery at the border between the renal cortex and the renal medulla. Now, several interlobular arteries arise from the arcuate artery. Look at here. This is the arcuate artery. You see, several interlobular arteries arise from the arcuate artery. Then several afferent arteriole arises from the interlobular artery. This afferent arteriole coils around itself and form a tuft of capillaries called glomerulus. Look at this is the glomerulus. This is the afferent arteriole which coils around itself and forms this glomerulus, a tuft of capillaries. Then this, after formation of this glomerulus, the vessel continues as the efferent arteriole. This is the efferent arteriole. Look at, here is the efferent arteriole. Afferent arteriole, afferent arteriole. This is the efferent arteriole. Now look at this model where you can clearly see that the diameter of the afferent arteriole is larger than that of efferent arteriole. That means the amount of blood getting inside the glomerulus is more than the amount of blood leaving the glomerulus through the efferent arteriole. This difference creates a pressure in the glomerulus. Under this pressure, fluid part of the blood with metabolic waste are pushed out the capillaries into the Bowman capsule space. Now here is the glomerulus. This is the glomerular capsule or the Bowman's capsule. Bowman's capsule has a visceral layer that covers the glomerulus, and its outer layer is the parietal layer. Between two, you have the space of Bowman capsule where the filtrate is accumulated. Now, glomerulus and surrounding capsule is called renal corpuscle. So this is a renal corpuscle. This renal corpuscle has two poles, as a vascular pole and a tubular pole. Vascular pole has an afferent arteriole which enters the glomerulus and efferent arteriole which leaves the glomerulus. At the tubular pole, proximal convoluted tubule initiates or starts from the tubular pole. Now, this efferent arteriole supplies tubules in the cortex. So those capillaries are called peritubular capillaries because they supply the tubules in the cortex. So this projections of these efferent arterioles are called the peritubular capillaries. Some of the efferent arterioles course down the medulla in a straight fashion. So the straight arteries running deep into the medulla are called vasa recta. We have two different types of vasa recta based on their origin. If vasa recta originating from the efferent arteriole, in other words, if the vasa recta is the continuation of the efferent arteriole, as you see here, this is called vasa recta spuria. Now, if vasa recta originating from the interlobular artery, as you see here, or originating from the arcuate artery, which is not illustrated here, but also there are vasa recta originating from arcuate artery as well. So the vasa recti originating from either the arcuate artery or the interlobular artery are called vasa recta vera. Both vasa recta are important, or they function in concentration of urine. Now, let's look at this corpuscles. This is the renal corpuscle. Renal corpuscle, as I have earlier explained, consists of glomerulus and surrounding capsule, or the Bowman's capsule. Look at here. Renal corpuscle plus renal tubules, these tubules, together make up a nephron, which is the functional unit of the kidney. And each kidney has 1,250,000 nephrons. And each nephron consists of renal corpuscle plus renal tubules. These renal tubules include the proximal convoluted tubule. The name proximal is based on the glomerulus. You see, it's starting from the glomerulus. So this is the proximal convoluted tubule, which continues with the loop of Henle. Loop of Henle has a descending line, which has thick part, thin part. Here is the ascending part of the loop of Henle, which has, look at here, a thin part and a thick part. Then here you have the distal convoluted tubule. Distal convoluted tubule joins the collecting duct through this cortical collecting duct. This is the cortical collecting duct. Several cortical collecting ducts unite each other and form a papillary duct. Papillary duct is larger one, or the duct of Bellini, which drains into the minor calyx, because minor calyx caps the tip of the renal papilla. Now look at here. Here we have two different types of nephrons. This is the cortical nephron, which is characterized with a short loop of Henle. And cortical nephrons are located outer 2/3 of the renal cortex. Around 85% of the nephrons are cortical nephrons. Now look at this one. This is located inner 1/3 of the renal cortex, at the renal cortex and medulla border. So this is called the juxtaglomerular or the juxtaglomerular nephron. Juxtaglomerular nephrons form around 15% of the entire nephrons, and they are characterized with a long loop of Henle. These juxtaglomerular nephrons also function in concentration of urine. So there are two anatomical entities which function in concentration of the urine. These are the vasa recti and the juxtaglomerular nephrons.